Welcome grade 12s. Today we're going to be looking at the revision of the DNA and the RNA, the nucleic acid, deoxyribose nucleic acid and ribose nucleic acid. We're going to start off with doing a few of the questions from the section A, which is the multiple choice question and the shorter questions of the paper. Right, remember when it's multiple choice, you have an answer sheet where you have to indicate just the letter. Please, all right, it says to you, if you have a look over there, just the letter. You mustn't write out the answer, just the letter that corresponds. If we look at the question, right, when you get the RNA and the DNA, what I would maybe suggest that you do before you answer the paper is just to write quickly RNA, okay, and DNA, and just write at the top of your paper that we're going to find C and G, and we're going to find C and G, cytosine and guanine, all right? And in RNA, we're going to find adenine and uracil, and DNA, we're going to find adenine and thymine. If you remember that little thing, just to put on the top of your paper, you're going to see as we work through the questions that it's going to be very helpful to you because lots of them are repeats of being able to know the different sequences that these appear in. Okay, first question. RNA differs from DNA. So already they're saying to you that RNA is different from DNA, and they want to know how is it different. Now have a look at each one, and what I would suggest is if you know it's incorrect, why don't you just put a cross through it and then go through the ones if you're not too sure if it is right or not. Okay, let's read to the first one. Has thymine and a phosphate group. Already, all right, if you have a look at that question that we did over there, it does not have thymine, so it cannot be A. Has a deoxyribose sugar and cytosine. DNA, all right, so it cannot be deoxyribose. The reason being RNA is a ribose sugar, so we know it can't be B. Number C is a double-stranded molecule. If we looked at our differences in RNA and DNA, one of the first things you would have said is that RNA is a single strand, whereas DNA is a double strand. So there we go. All right, which leads us with the last answer, number D, has uracil and a ribose sugar. And if you have a look over there, all right, I did mention, yes, it has uracil and ribose. Remember, RNA, R stands for ribose. Okay, so the answer that you should have had, there we go, is number D. Okay, let's go on to the next question. You're going to notice this next particular diagram. I want to just see if I'll show it to you over there. This is a very popular diagram that they tend to ask in the paper. It comes a lot. It can either be in the multiple choice questions or it can be in the longer questions. You're going to come to recognize this diagram quite a bit. Okay, the question here is, okay, there is based on the diagrammatic representation below of a part of two different nucleic acids. They are telling you something there. They are telling you that in this diagram there are two, so you should know already, all right, that one is going to be RNA and the other one is going to be DNA. So they've already said to you two. All right, found in the cells of organisms during a stage in the process of protein synthesis. All right, and that's something we're going to look at later. Let's look at molecule 1 and molecule 2. Now, if I go back to that when you wrote at the top of your page, again, what does RNA have? Keep on reminding yourself. RNA is C, G, A, and U. What does DNA have? C, G, A, and T. That is the most important thing that you need to know when it comes to, all right, to the DNA and the RNA. So if we have a look over here, all right, what we should notice already is that we know that one is RNA and the other one is DNA. They look exactly the same, but the bases, all right, remember, the bases are going to give you a clue. Look what I said, RNA has got, all right, uracil, and DNA has got thymine. If you have a look at the two sketches, you will see that molecule 1 has a T base, which is thymine, and molecule 2 has a U base, right, which is uracil, which you should be thinking now, okay, that would then be RNA, and that would then be DNA. Okay, so let's carry on with the question. All right, the diagram above illustrates the process of. Now, even though this whole thought process is going through your head, what is it showing you? Okay, it says the process, and when you did the processes, there were 3 names that you needed to remember. Replication, which was when DNA makes a copy of itself. All right, transcription. Let me see if we can get it here. Transcription is when DNA, all right, gives rise to RNA. Okay, and translation is where we have making of a protein, all right, and mutation is where something goes wrong. All right, so if I had a look at those 2 molecules again, all right, I know it can't be number A because DNA replication means there must be 1 strand of DNA. We know that there's not. There's an RNA. Number B is transcription. All right, cracking the code. And that's making a DNA from an RNA from a DNA. All right, and that would then be my answer, because you can see already, because translation, you would need to find things like tRNA, okay? Right, and you would see words like anticodon, etc. So already from that picture, you can see what they expect of you. Okay, going on to the next question. That question was, they gave you the 1 diagram and they actually asked quite a few questions on it. So if you knew the 1 diagram, it's actually quite nice if you understand the diagram that quite a few questions were related to it. All right, so we're going to go here. The process illustrated above occurs in, and we already said that the process was transcription. All right, and where does transcription occur? Transcription is when the DNA and the RNA, all right, are in the nucleus. Remember, the DNA cannot come out of the nucleus. So if the DNA is involved, the DNA is always inside the nucleus. So your answer then would be number D. All right, remember, the DNA is always, all right, kept to housing inside the nucleus. So if you have a DNA picture, it cannot be the cytoplasm or outside the nucleus in the cytoplasm. Okay, let's go on to our next question. All right, it's the same diagram again. As I said to you, same diagram, but it can ask you a multiple of questions from that diagram. Now it's asked you, as I said, I'm going to quickly go back to, all right, this page that we had over here. There we go. As I said, now it's going to take into account the differences between RNA and DNA. So if you had written it on your diagram, all right, then you should have had the answer in front of you, because it asks you an observable difference, all right? And it's 2 things. What is the difference between the molecule 1 and molecule 2? They're not asking you to, all right, give the difference that comes that it's double-stranded or something. The key word there that they asked is observable. In other words, from the diagram, what can you see? Right? What is the 1 thing that you can see from that particular diagram that is going to apply to this question? Let's go through the answers. Molecule 1 is double-stranded and molecule 2 is single-stranded. Now remember, what did we say molecule 1 was? Was DNA, all right, because it had thymine, and molecule number 2 was RNA because it had uracil. So molecule 1 is double-stranded and molecule 2 is single-stranded. Is that correct? Is the observable difference? The answer is right if you weren't given a picture, all right? But they want to ask you, what can you tell from that diagram? And you can't see, it doesn't show you that either of them are double-stranded. That's a bit of a tricky question, and it's your first option, so you do tend to go for A. Number B, molecule 1 contains deoxyribose sugar. Yes, it does. All right. And molecule contains ribose sugar. Yes, it does. But if we have a look at the diagram, it doesn't tell you that. All right. The same circles, here we go, the same circles are shown and they don't show any difference. So it's not an observable, all right, observable difference. Again, you see how tricky that's going to become. You must read your questions very carefully. Number C, molecule 1 has thymine and molecule 2 has uracil. Let's go to the picture. Molecule 1 has thymine, and there we go. Molecule 2 has uracil. Can I see it on my picture? Yes. All right, which means that number C is correct. Let's just have a look at number D just to make sure. Molecule 1 is longer than molecule 2. If you look at the diagram, you will see that there is no difference in the length of them. So that also then has to be incorrect. Okay, shall we go on to the next question? All right, multiple choice is always so, well, so nice and easy, but it gives you a bit of an idea of the words and the phrases that they're going to use. It's a bit of a warm-up exercise for the rest of the paper. Okay, the next question, all right, and you're going to see again, very important, as I said to you, have at the top of your paper what proteins do I find in my RNA and what do I find in my DNA, the nitrogenous bases. Now, the next question, the mRNA, all right, now you need to understand that you've got the DNA, I'm just going to write it here. DNA gives rise to mRNA, all right, and then mRNA, right, is going to join with tRNA. I just want you to keep that in your mind because it's important. If you know the codes for each of those, one of those, you can always find the codes of any of the other two. Okay, now they've said the mRNA sequence from a portion of a DNA. All right, so it's from the DNA. The template is GATCAAS. Now have a look at the question. The mRNA sequence from a portion of a DNA template is, all right, what is the answer? Now you take your pencil and you write under there, they are partners, okay? They want to know what the mRNA sequence is. They've given you the DNA, they want to know what the mRNA is going to be. Now remember, C goes with G, that's always. Here's always the tricky one. Remember, when it comes to RNA, all right, we have got A and U. Adenine and uracil. So my A goes with U, my T goes with A, all right? Now you also know it's a DNA because it's got thymine in it. C's partner is always G. Here, A's partner is always U on the mRNA. So if I have a look, that does not match, all right? Okay. That does not match, does it? Yes, it does. All right, so what do we have? So we said that doesn't work. Have a look. C, there we go. U, A, G, U, U. There we go. The answer is number B. All right. Be very, very careful. Lots of you are going to fall into the trap of number A. The key was it must be the mRNA code. Please remember that A, adenine, all right, very big. Adenine, all right, must always become uracil. Adenine and uracil are partners from the mRNA and onwards. Okay, that is very important. It can cost you if you keep on putting thymine with adenine in the RNA. Thymine and adenine are partners on DNA only. All right, let's go to our next question. Again, you will see the questions are very similar to each other. So if you know your bases, your complementary bases, then this section is going to be quite a breeze for you. Okay, the next question is the nitrogenous base. And remember, what are our nitrogenous bases? Again, cytosine, guanine, adenine, thymine, and uracil. All right, which replaces thymine here? It's the same thing that I've been stressing to you all along. It replaces thymine in a RNA molecule is. Right, so in other words, which nitrogenous base is not found on the RNA, but only found on the DNA? And what did we keep on saying? All right, uracil. There we go. So uracil is my answer. Remember, uracil is only found in RNA. Thymine is always found in DNA. Don't get those two confused because the rest, C and G, are partners for life. Cytosine and guanine, you can't make a mistake there because they always go together. They don't ever stray from that. Okay, let's look at our next question. This can also be quite a tricky question because you look at it and the first thing I thought, and I think some of you also thought, is how do I know which percentage, right? I don't know how many the percentage of each of the bases, but have a look. The percentage, all right, doesn't matter about the percentage. It matters what percentage matches the other one, and that is the clue. Let's have a look at the question. During an investigation, the DNA of an animal cell was analyzed in a laboratory, and the results are shown in the table below. Which of the following is a correct identification for the bases called X, Y, and Z? Now they've given you a clue. The first clue is that if they didn't give you one of the bases, then you wouldn't have been able to work it out. But they give you a hint, and they say that there's 20% in adenine. All right. Now, they've also said DNA. That is your hint. Always know, is it RNA or DNA? 20% in DNA. What is its complementary partner in DNA? There again, have your little pen or write your little rhyme next to it. Okay, A goes with T and C goes with G. So we can assume that if there's 20% of adenine, obviously there must be also 20% of thymine. All right, so there we go, 20% of adenine, 20% of thymine. So now they've given us a hint, all right, because we know now that Z must be thymine because they're the same bases, they must have the same partners. All right, so let's look at our A, B, C, and D. Look at Z first because that's going to give us what it is. So we know that it's not that. So B is out because it's not cytosine. And we can already take A out because we know it's not adenine, which leaves us with these two. Now, they don't tell us which is X and which is Y. So you don't know which then would be cytosine and which would be guanine. But let's have a look at the options that they give us in the multiple choice. If you have a look, number A, they tell us that that's cytosine and that's guanine. All right, could that work? Yes, it could, definitely. Now they tell us that X is guanine. Yes, it could be. But they tell us Y is adenine. It cannot be adenine because they've already said that adenine has 20%, all right, not the 30%, and you can't have all the different double groups of adenine. So just from that one little label makes the whole of D invalid. So your answer, all right, would then be number A. Just by looking again at the corresponding, all right, at the corresponding numbers. This question is also, it's a bit of a tricky question because some of you, all right, might not read it properly. If all 18 nucleotides, now remember again, here's a term, nucleotide. What is a nucleotide? A nucleotide is, remember, the sugar, the phosphate, and then one of the bases, all right? So C, G, A, or T. All right, because they tell us that it's a DNA strand. They say if all nucleotides of a DNA strand code for amino acids, all 18 nucleotides, all right, code for an amino acid. How many amino acids will be present in the polypeptide that is formed? All right, so if all 18 nucleotides of a DNA strand code for amino acids, they're saying each one of them, all right, has a code for amino acids. How many amino acids will be present in the polypeptide that is formed? Now, I think most of you, right, would answer that it's going to be 18. 18 nucleotides, therefore 18, all right, therefore 18 amino acids. But the question was, all right, how many amino acids will be present? Now, if you think of how transcription occurs, one thing that you would have learned is that when they come from the DNA, all right, they are in codons. They are in bases of 3. Okay, so we're going to have 1, 2, 3, 1, 2, 3, 1, 2, 3, and one of them, all right, codes for one amino acid. One of them codes for another amino acid. So it's actually 18 divided by 3, and your answer should then have been 6. All right, I know lots of you would have gone for number 18, number B, but remember, all right, when we find the amino acids, three bases, all right, that makes up your codon, is going to be, all right, need to find its tRNA, and the tRNA has 3, right? Remember, 3 anticodons on it. So we group it into those codons, or some of you might recognize it as triplets, right? Sometimes they use the word triplets. Okay, guys, we're going to go for an ad break now, and then we'll be right back. Welcome back, grade 12s. We're going to carry on now with our revision. We're now going to look at the questions from the longer questions from questions 2 and 3. Okay, I'm not going to remind you again about the partners. We've done it a lot in the first segment. I'm now going to assume that you now know that all of them, which they match together. All right, let's start. The question is, study the diagram below which represents a part of a nucleic acid molecule and answer the questions that follow. Now, they haven't told you that it's DNA or RNA, so from the diagram you must be able to see that. So the first question, identify the nucleic acid in the diagram above. All right, if we have a look here, what you should notice, first of all, it's double-stranded because there are the hydrogen bonds. All right, that is your first cue. So if it is double-stranded, all right, then it must be a DNA molecule. That's the only information. It doesn't show you anything else, but it's double-stranded, right? So you know it's a DNA molecule. Okay. Then they ask you another question from that picture. They want you to label the parts 1 and 2 and nitrogenous base 3. All right. Okay, number 1. Number 1 is, I'm going to label on here. Number 1 is a sugar. All right. Now, the reason why I always remember that, a sugar, it's stuck to everything. And if you know, if you eat too much sugar, it always sticks to the hips. All right. So always remember sugar sticks, because often you guys get confused. Okay, this little one is phosphate. All right. And then they asked you for number 3. They gave you adenine. All right. So we know that its partner must be thiamine. So if we were to label it, part number 1, we said was the sugar. All right. Part number 2 was the phosphate, and our nitrogenous base, number 3, was thiamine. Okay, let's carry on with the next question. All right, again, same question, all right, from the paper. Now it asks you, what is the collective name, all right, for the parts numbered 1, 2, and 3? 1, 2, and 3 are a building block, all right, a piece of Lego for when you build the molecule. And I said just now, I gave you the answer. When we have a sugar, a phosphate, and a nitrogenous base, what do we call it? A nucleotide. All right, remember a nucleotide, a sugar, a phosphate, and a base. They all join together, they're the building block, and they're going to make a nucleotide. See, nice, all same questions, all based from the one question. Now, this next one I found was a bit tricky. You need to be able to read it carefully. It says, describe the role of DNA during transcription in protein synthesis. They did not ask you to describe the process. They said, what must DNA do during this role? All right. First thing that DNA must do, what must it do? All right. It must be able to unzip. All right. And we know that it does that by enzymes. Second of all, what must it be able to do? You see, it's 4 marks. 4 marks. That means you need to have 4 facts. All right, look at the mark allocation. Unzip the enzymes. What else must it do? It must be able to form a template. All right, it must become a template, because when it unzips, all right, what does it make? It makes that DNA open for the mRNA. So there we go. It's a template for mRNA. Believe it or not, those two are 2 different marks. So just by saying that, you will get, all right? And then the last thing that it's going to do, where must it stay? And I said that earlier, it must stay in the nucleus. Okay, again, the question, what is the role of DNA? They did not ask you to describe the process of translation and transcription. Okay, let's go on to the next diagram. This is the next question, right? And as you can see from all our little codons, again, a DNA and RNA question, that the different kinds, if you noticed, you're not going to get all of them in your paper. I'm just showing you the different variety in which they can ask you the questions on DNA and RNA. The diagram below shows a sequence of nitrogenous bases of a small strand of DNA. All right, there we go, DNA. They've told you that it's DNA, which codes for parts of a protein molecule. Write down the mRNA codon sequence that reads from the left, all right, from the left to the right. So you must go from here to there. Now, it is DNA. They want the RNA code. C always goes with G, okay? G always goes with C. G always goes with C. T goes with A, and there. A goes with U. It's on the RNA. T goes with A. C goes with G. C goes with G, right? And T goes with A. So there is your codon sequence. You would write there G, C, all right, C, then you would write there A, U, A, and then you would write the third one, G, G, A. Each of those would be worth 1 mark. Okay, all right, remember how important knowing the bases, which ones are complementary bases. Okay, the next one is quite a tricky, looks tricky, but if you read the question carefully, it's actually quite a nice little simple question. But once again, understand the question properly. All right, they've given you a table with the different anticodons for tRNA are going to have different amino acids attached to it. So if you'll see over here, what they're trying to say, now this is very important, there's your key. They're telling you that the tRNA, remember, the tRNA comes in, it has an amino acid attached to it, and then it's got 3 bases. So those 3 bases together give a name of an amino acid. So let's have a look what the question asked us. The question says, select and write down from the table above the amino acids in the correct sequence. Okay, that would be required for the base sequence of mRNA shown below. Now remember, they're asking you, this is mRNA. You must then convert it to tRNA first so that you can then find the correct name. So don't use these ones, because if you use these ones, then you're going to look there and you're not going to know what to do, because they're not going to match. So let's go. GGG would be C, C, C. C is G, C is G. A is U. Remember, it's not DNA. A is U, G is C, and U would be A. Okay, so let's have a look now. Go to our table and see if we can find an amino acid that's CCC. All right, there we go. If I have a look over there, CCC, and I see that the amino acid is called glycine. All right, so I write down glycine. Because remember, they said you must write it in the correct sequence. You mustn't get them all confused. So that is glycine. Then we go to our table. We're looking for a GGU. All right, let's have a look. We look. Can we find anything? Aha, there we go. GGU. All right, and that is called Prolene. So what do I write next to it? Prolene. Doesn't matter if you've never heard the names before. That's not the point. It's just, can you work out the code and then find the corresponding name. So you've earned 1 mark, you've earned 2 marks. Our third mark, let's look. UCA, I go to the table. Oh, there we go. There's UCA, and what's its partner? Serine. All right, so what do I write next to it? Serine. There we go. 3 marks. Right, for 3 marks, 3 amino acids. I might not necessarily have known the names. That is not important, but I do know the codes, and that is the most important thing that you need to know. We're going to take a short little break quickly, and then we'll be right back. Welcome back from your short little break. We're now going to go into the last segment, right, of the revision of DNA, and I think it's the one that most of you dread the most. I'm going to look at an essay question on DNA and RNA. Right, now the essay questions have moved away from opinion-based questions, and you'll notice that it's very factual. So it is possible, if you study hard, that you'll be able to write the essay nicely. Okay, let's have a look at the question first. The question says, describe how proteins. Now remember, we had a previous question that just said describe the role of the DNA. Here, they actually want you to describe how proteins are formed. All right, so what do they want? They want translation. And how do we get there? All right, we have to make an RNA. They want transcription. All right, those are the first two ones in a cell. Then they add an extra part. Okay, and this, I think, of all the ones is a little bit of the tricky one, but I'm sure you guys will be able to manage it. It says, and explain the impact of two types of gene mutations, all right, on the formation of proteins. So in other words, what can go wrong during the transcription and during translation that is going to bring about a fault, all right, in the coding. And there, they just want you to mention two. So if we have a look what they are looking for, they want transcription, which is going to be one paragraph. They want you to explain translation, which is going to be your second paragraph. And then they want you to give examples of two gene mutations. That is going to be your third paragraph. If you will notice, they are looking for 17 facts, right? 17 points on content, and then you will notice the synthesis is for 3 marks. So what they do there is if your essay is very well written, if it looks like you know your work, those kind of things, they'll give you 3 marks. Then if you sort of know your work, but there's a little bit of inconsistencies, they'll give you 2. If you've missed the point and there's not too much that you've added there, they'll give you 1. And if obviously you're going to get 0 if you don't write an essay or you have missed the topic totally and none of the points are relevant. So you need to have 3, right? You need to have 3 paragraphs. I know for some of you it's difficult to write in paragraphs. Okay, so formulate, if you have the time, write in point form first. Okay, just put in point form, and it's much easier for your marker to actually see what you're trying to see as well, right? Especially if paragraphs frighten you and the volume is just too much. Just jot down the points, and we're going to show you the memo as well, and that's also jotted down in point form, which is much easier for the markers to mark, right? You might lose points in synthesis, but you rather attempt the essay than to leave it out, because that's 17 marks and plus the 3, 20 marks is a lot of marks on your paper. Okay, so let's start having it. I'm going to have the answers on the side because you will see then how what the markers are going to look for, but I'm going to explain the process with diagrams as well. The first paragraph we said, right, was transcription, and that is when, all right, we have a DNA molecule, and the DNA molecule acts as a template to make a mRNA molecule, messenger. Right? Remember, the DNA is too big to fit through the nucleus. So what do we want? We want the DNA, right, to do all the coding in the nucleus and taking that message and then taking it out to the cytoplasm onto the ribosome. Right? So you need to make sure when you talk about it, you are specific. You need to be biological, right? And you can't waffle here, because if you waffle, they're not going to give you the benefit of the doubt. Right? Describe it step by step. Right? That's how you should have actually put it out in your study notes. Step by step by step of what happened. So let's go. Here's the steps next to me, and we're going to go through them. Okay. Remember transcription. What do we have? We have the DNA. All right, I'm going to just make them a bit further away from each other. All right. The DNA is a double-stranded nucleic acid. Right? And what do we have on the sides? Now, you need to think of words. All right. You must be scientific. So we say the DNA unzips. All right, because we know the DNA was joined and then it unzips. Now, when it unzips, tell me, how did that happen? enzyme action. You don't necessarily need to know, all right, the polymerase, the enzyme, just as long as you know that it's unzips by enzyme action. If you have a look, you've got a mark for double-stranded, all right. You say it unzips, and you could have added in there. Notice, all right, you had to have 6 points. And if you count all the ticks, there's multiple amounts of points that you could have used, as long as it's in sequence. You can't tell me that it unzips right at the end, all right? Remember I said your essay must be logical, okay? So what happens first? First thing, the DNA unzips. Enzymes unzip the DNA. Now we have 2 strands of DNA that are open. All right, and as I say to my kids, these don't like to be alone. All right, as soon as it opens, all right, they need to form partners, and that's exactly what happens. Floating in the nucleus, all right, in the nucleoplasm. Now listen to the words: are complementary RNA nucleotides. Okay, I'll say it again. Complementary, all right, nucleotides. Complementary means they're the bases that are going to fit to the template part of the DNA. Okay, so you've got that. So having a look here, using free RNA nucleotides, that's another word for complementary. They're floating there. It's like when you build Lego, all right? If you want to make something and you've built something you want to add on, you just go into your Lego box and you take it out and you carry on building. That's exactly what it is like here. There's little building blocks, the nucleotides, previous question, and they then are going to attach themselves to the DNA. Okay, now remember they're a nucleotide. So if they have a nucleotide, they're going to have a sugar, they're going to have a phosphate, and a base, all right? They're going to have the complementary bases. And remember, RNA, we have C, we have G, we have A, and we have U. And then in DNA, what do we have? C, G, and A, and T. All right, so let's have a look how it should have been said. The coded message for protein synthesis is thus copied onto the mRNA. This is the DNA. These might just be letters, but those letters, remember, transcription means a code. All right, making a code. The DNA, they have got how do you make a protein. They now need to get that message from the nucleus out into the cell where the proteins can then be made. Okay, so do you see the process? You need to make the message makers in the nucleus. That message now needs to get out of the nucleus into the cytoplasm and onto a ribosome. Okay, so now you should have had, look at the words, double-stranded, unzipped, hydrogen bonds break. You can say the complementary basis, you form an RNA molecule, all right? It carries the code to make protein synthesis. And as I said, what is the third part? There it goes. Once it's complete, remember, mRNA is small so that they can fit out of the nucleopores in the nucleus. All right, so what happens? We call it a messenger RNA because it's carrying a message. It's carrying the codes, all right? And where does it go? Out into the cytoplasm and onto a ribosome. That is the first part, transcription. 6 marks there, as was the indication, all right, by the memo, 6 marks. But if you have a look at the text, you will notice that there is room, all right, for, don't only have these, what you write, if you don't have every point, you're not going to take the marks out. You need to have the six marks, but you need to make sense, and it needed to be in order. If we have a look at translation, all right, that is the next part. The mRNA now goes and sits on a ribosome. Now remember that mRNA is in codons, okay? Now, if you have a look over there, there we go. They talk about the codons of the mRNA. But what are they looking for? In the cytoplasm, all right, are tRNAs. I always call them taxis. All right, T for taxi, because that's exactly what they do. All right, each of them are carrying an amino acid. So now the bases again must match. So if this is CCG, all right, that will be GGC. So the matching anticodons, they come and they land onto, they join, not join, but the complementary bases again. Complementary bases then will lie next to each other. Now, if you've got amino acids, I always say it's like making a kebab, all right? So you take a stick, here's your pieces of chicken, you take a stick and you join them all together. You had little pieces of chicken, now you've got a kebab, all right? That's exactly what happens. What do we have? A peptide bond, all right, is formed, and when that peptide bond joins all the amino acids together, all right, what do we have? We have a protein. All right, so if you have a look at the model answer, it tells you that tRNA, there we go, has its amino acids, all right, they attach to the RNA. We had that the anticodons match the complementary bases. We had that, all right. The amino acids join, very important, by peptide bonds, and then a protein is made, all right. Now remember, taxi doesn't just disappear. These taxis will then, tRNA, will then go and pick up another amino acid to make another protein. Okay, so far we have got 12 marks. Now we're going to look at the last part, all right? And they asked you there, what happens if something goes wrong? Now, first thing you need to know, you must tell what is a mutation. So the first thing before you write, all right, a definition for mutation. So you say that during the DNA replication, all right, something went wrong. When DNA makes copies of each other, something went wrong, okay? So here, one of them, point mutation, replacing one base of a codon with each other. All right, so what happens if C joins with A? All right, what happens if C's here and all of a sudden we add in another one? All right, so mistakes can be made. You guys make spelling mistakes. You put the wrong letter where it should be. You add in letters where it shouldn't, or you leave out. All right, but if that happens on your DNA, that's a mistake. That's a mutation. All right, it's a small change, as you see here. It's a small change that can result in a different amino acid that's going to be formed, which can have, right, some serious repercussions. So if we looked at it, we get the point mutation, all right, where we replace one. Sometimes you get shift, where one is replaced, one is put in, we add in one, we leave one out, all right, we put the wrong one there, that is the mutation, all right. So then what happens? Our sequence is wrong. Our sequence is wrong. We're not going to be able, right, to all the joining again. What's going to happen is there's not going to be a partner for it. So some mistake is going to occur, all right? And then if you make a mistake in your protein, then you've got a protein that's got a function that is totally different from the one that it was meant for. It might be a serious mutation, all right, or it might be slightly insignificant, but it's a mutation nonetheless. So just to recap, there we go for your essay. Right, please, you can write it out in point form if you're not feeling confident, but if you have the time, take those points, put them into paragraphs. Be clear. Some people say don't write headings, right? If you have a look over here, you can start off by saying transcription if you're taught not to write headings. It just makes what you're writing underneath it a bit more clearer to the marker. All right, and I think that is all that we have time for. Wish you all the best for your exams, and goodbye from me.